So thank you for joining us for episode 4 of Up to Date COVID edition. We're really excited to have Dr. Tiffeny Smith here for our rapid diagnostics perspective. And Paula has been posting the pre-discussion survey form within the chat. So if you could go and take that survey super quickly, it'll only take like about 30 seconds to fill it out. And I think we'll go ahead and get started. So Dr. Tiffeny Smith is an infectious disease specialist at Genmark Diagnostics. She earned a Bachelor of Science degree in microbiology and nursing from the University of Oklahoma and a Doctor of Pharmacy degree from the University of Oklahoma College of Pharmacy. She completed a PGY1 in pharmacy practice and a PGY2 in infectious disease residency at the Johns Hopkins Hospital. And prior to joining Genmark Diagnostics, Dr. Smith practiced as a clinical pharmacy specialist in infectious diseases and antimicrobial stewardship, most recently at UT Southwestern Medical Center. So we're very excited to have her here today, and I'll let you take it away.
Thank you so much, Michelle, and thank you, UIC College. I joined Genmark about almost a year ago, and Genmark Diagnostics quickly recognized that a relationship not only with the laboratory but also clinicians is really important, especially when it comes to implementing molecular rapid diagnostics. And you can go to the next slide, Michelle.
Yeah. Sorry, I think you froze for a little bit earlier there, so I couldn't hear you.
Oh, okay. No, that's okay.
I think it's fine now, so I think everyone can hear.
Okay, perfect. And so Genmark quickly realized that it was important to have a relationship not only with the laboratory, but also clinicians when it comes to molecular rapid diagnostic testing and implementing into a hospital or institution. And so Genmark Diagnostics hired me as an infectious disease specialist, a clinical role, to help create and deliver presentations to different audiences that usually include infectious disease physicians, pharmacists, infection preventionists, and even hospital administrators. And these presentations are really designed to demonstrate the clinical value of molecular rapid diagnostic testing, as well as provide some economical value to the institution as well. Another part of my job is to assist institutions that are adopting rapid diagnostics to help them implement it clinically as well as within their stewardship program. As well as internally, I help educate and train our sales organization to help them feel more confident in meeting with clinicians and really being able to speak their language and understand where they're coming from. And lastly, another part of my job that I absolutely love is that I'm able to interact with other pharmacists, physicians, as well as infection preventionists to build these relationships with them and really gather their opinion and their thoughts on rapid diagnostics, and even bringing some of those ideas back to the company to develop, to expand and develop their infectious disease portfolio. So enough about me. That's just a preview of what I did prior to this pandemic. But now, during the pandemic, a lot of hospitals are limiting visitors, obviously, right? So industry meetings face-to-face have been very limited ever since early March. While I'm based out of Dallas, I'm not geographically limited on who I meet with. And so I've been trying to do a lot more customer calls via Skype or GoToMeeting, taking conference calls, and really taking this time to also expand on our internal education and training, as well as update process documents that have been sitting on my desk, and also learning, right? I'm a lifelong learner, and this emergency use authorization of diagnostic testing is pretty new to me, coming from the clinical side, and understanding the regulations and how institutions are able to bring on a test that's considered emergency use authorization is something that I've been working on. And one of the things that I did as a pharmacy student resident, even as a practicing pharmacist, is when I had a question about something, I would write it down in my notebook, and then I would go back and look it up. The difference here is I don't have a preceptor that's going to ask me about that question. So I'm really taking the time to go back and answer some of these questions and look it up, because my first 9 months with the company was such a steep learning curve coming from a clinical side to industry that I had a lot to learn, and I didn't always have the time. So really being able to learn and develop during this time, to also be supportive of my team, is really important to me. So I'm going to switch gears a little bit. Enough about me. I'm going to talk a little bit about our instrument. Go ahead and do one more click. So there's an image at the bottom. Yep. I think it's really important to highlight a little bit about our instrument. The main focus of today's talk is going to be about our SARS CoV-2 assay. However, there are some things that I want to cover that I think is really important when it comes to diagnostics. So Genmark quickly realized that they wanted to develop a solution to conventional molecular testing. They want to develop a sample to answer solution with a rapid turnaround time, ultimately improving patient care. And molecular diagnostic testing really began to take off in 2009 with the influenza H1N1 outbreak. And since then, conventional approach to molecular diagnostics is sophisticated. It has a lot of hands-on steps, as demonstrated here in this image at the top, taking anywhere between 6 to 24 hours to process the sample. And it's really because there's so many steps, and these steps are complicated. So think back to your biomedicinal chemistry, right? PCR requires many steps, and it's very labor intensive. So how can we develop a solution into patient care where results should be rapid and the workflow should be pretty simplified? And that's how the ePlex came to light. Several years ago, we met with customers, had focus groups, asked questions, really to learn about the current products that were there and what their limitations were, and really try to develop a system that improved those limitations of other instruments, as well as provided solutions to the lab, starting with how they process a sample, how the information goes into their laboratory information system, quality control testing, even as far as getting the results to the patient into the EHR. And that's how we came up with the ePlex instrument at a high level. Okay, next slide. The picture on the previous slide was a little small, so I wanted to show this. This is our actual instrument, and how you can all think about it is, think about a glucometer, right? As pharmacy students, you guys are taught to work with glucometers and test strips and read blood sugars, right? And so this instrument is really the glucometer, right? It is where the cartridge goes in to process a sample. But what I really want you all to focus on is there's 3 components to this instrument. The base, which is that middle part where there's a touch screen. But on the sides, there's a tower. And those towers have what we call bays. And each of those bays, there's 6 bay to 1 tower, is where the cartridge would go into to process the specimen. And it's very similar, like a test strip going into a glucometer. Usually glucometers and test strip can only process 1 strip at a time. But with ePlex, you can actually process up to 6 specimens in 1 tower. Because the bays are completely independent. And I think that's something that I didn't really appreciate as a practicing clinical pharmacist. I responded to vancomycin levels and aminoglycoside levels, and I could get an idea of if when the level was drawn, when I should get a result back. Well, in this scenario, results are usually done off of the machine within 90 minutes. So when the lab tech loads a sample, you get an answer within 90 minutes. But if you have a lot of samples, such as when you're in the middle of a pandemic and there's 2,000 respiratory tests that need to be ran per day, and you can only technically run so many tests, there's going to be a delay in getting those results. And I think that's something that I didn't really appreciate when I'm trying to look for a result so I can make a decision and there's a delay. There could be a delay because of changes in volume, right? And so that's one aspect of this big picture that I wanted to point out. Next slide. And to talk a little bit about our technology, I won't get into the nitty-gritty of it, but basically our cartridge, which is this image at the top with the green label, that is our cartridge. So equate that to a test strip, right? And this cartridge contains all of the reagents and the technology that's necessary to carry out the PCR and detection. The sample is loaded into the cartridge. It already has the reagents. The nucleic acid is extracted from and purified within this cartridge, and the e-sensor technology detects a target DNA. And I just wanted to point out that all of that is just done in this cartridge. So next slide. And so the workflow, I think this is a great depiction to give you all an idea. I don't know how much time you guys have spent in the lab and been able to see this in action. Once the sample is received, it can be dispensed into the cartridge, and then the lab technician scans the barcode and loads the cartridge. We've done studies, numerous studies, and on average, this whole process takes about a minute and a half, 90 seconds. It's pretty fast. So think about PCR, conventional PCR, and think about this. This is how we've really improved the process and developed the sample to answer that's very efficient. And do a couple clicks. Yeah. And so, as I mentioned earlier, these bays where the cartridge goes in, they're completely independent. So if one bay is being used for processing one patient sample, the tech can also put in another sample in an open bay. If a bay goes down, it doesn't affect the other bays. And this is really important, especially to account for changes in volume of samples coming through. The thing that I want to point out is, so ePlex, Genmark has 3 FDA-approved assays prior to the SARS-CoV-2 test. We have blood culture identification. We have 3 assays within that, and then 1 respiratory. And so the lab can actually process specimens that are respiratory or even blood culture all on the same instrument. And so that's also something that is unique, right? It's not like we can put an INR finger stick on a glucometer and process that. So I think that's something to appreciate as well. And it allows for laboratories to really adjust to changes in volume. But clearly we did not, we were not able to prepare for the demands during a pandemic. And I'll get into a little bit of that in a couple of slides. Next slide. The other thing, another feature that I wanted to point out about our instrument is that it's scalable. It's pretty small. It could be put on a bench top. The 4 tower is the largest. It has 2 towers on each side of the bay. It has each side of the base, and it has a total of 24 bays. And if you do the calculations, it can process about 288 samples per day. It takes about 90 minutes to run each sample. So you think about it, Chicago, where you guys are experiencing a COVID-19 pandemic, right? I read early March, Chicago was processing about 2,000 patient tests per day. And so do you really think ePlex alone would be able to process that many tests? No, right? And so what I wanted to point out is that most laboratories are going to have to think about, one, what other manufacturers can support my volume? Do I need to have multiple manufacturers within the laboratory to do testing for, let's say, critically ill patients in the ICU? Maybe you reserve ePlex for that. And then if you can screen patients or outpatients or even healthcare workers, then maybe using a larger scale instrument for those. I just think that given the situation that we're experiencing now, we have to be very creative in what we do. And unfortunately, that means thinking about how to change our testing algorithm and prioritizing the most critically ill patients and our healthcare workers, our first responders. And we didn't have to think about that in the last few years, and now we do. And so it's so important to partner with different manufacturers to really identify a solution that works for your patients. And lastly, so another aspect of rapid diagnostics, I think, is important for clinicians and future clinicians and clinical pharmacists to really identify and recognize is what are the targets that are being tested, right? So I want to talk a little bit about our blood culture panel, because this is the most commonly used right now. We have, under blood culture, we have 3 different panels broken up into gram-positive, gram-negative, and fungal. And within those panels, we're not only able to identify organisms, but also resistance markers. And it's super important to be able to do that, as it may affect treatment for some of our patients, especially critically ill. It's interesting, there hasn't been any data published because the information coming out is it changes so much about this pandemic, right? But one of the questions I am asking myself and asking my colleagues that I talk about that are still in the hospital is, these patients that come into the hospital, hospitalized, on the ventilator, on average, are ventilated for weeks, right? And they're in the ICU when they're ill with COVID-19. That increases their risk of hospital-acquired infection. So how many of these patients are having concomitant bacteremias or even fungemia? I want to point out on our fungal panel, we're able to identify Candida auris, which is a popular topic in Chicago prior to the pandemic. I think COVID has really outshined a lot of these conversations, but it's important to point out. And so I know we're not going to know that data, and probably until all of this is over, but it is something to be aware of, that we have assay that can be ran on the same instrument using blood culture for those patients that have a bloodstream infection. And the next slide. And I need to talk a little bit about our respiratory pathogen panel. So this is our multiplex respiratory panel. It has viruses, 15 viruses and 2 bacterial targets. This has been around and utilized. You'll notice here that the coronavirus that's listed here is not the SARS CoV V2. And so we quickly learned that this pandemic was happening. And so we developed the SARS CoV V2 assay, which is on the next slide. So just to highlight the SARS CoV V2 test, it comes 12 cartridges per kit, and these kits are stored in the refrigerator. So think about that. Like, does the lab even have enough space to store the cartridges or the reagents necessary to carry out the test, as well as how many instruments do they require and where are these instruments placed? And another key factor out of this test is that the sample type. The sample type that is approved for use with our test is a nasopharyngeal swab. It has not been studied to be used on a bronchial alveolar lavage or even a sputum, right? So that's really important to know as a clinician, because oftentimes you all may be interfacing with the nurse and giving these recommendations to make sure they're appropriately collecting the correct specimen for the test. The assay design is a dual gene assay, and I'll talk a little bit about that later, but it ensures the broad inclusivity of the SARS CoV V2. And so when I put together this talk, something that I thought about is, what should our clinicians and our healthcare providers know? A good resource is the FDA webpage that lists all of the emergency use authorization manufacturers. As of yesterday, there's 29 manufacturers that have EUA. And each of those manufacturers, they have their package insert as well as fact sheets for patients and healthcare providers. So if you want to learn more, I recommend going to that website. But for us, I think about these 4 questions. What do you need to know about the testing? First, you need to know that the specimen type is nasopharyngeal. This test's performance has not been studied in immunocompromised patients, and it hasn't been studied in patients that are asymptomatic, like say if you wanted to screen patients, right? Its performance has only been done using nasopharyngeal and in patients that had signs and symptoms of COVID-19. And what are the results? So the results are qualitative. It is positive or negative. It's not quantitative. It does not provide a numerical value associated with the virus. And what does it mean if the specimen tests positive for the virus? It means that the RNA was detected. It likely is that the patient is infected, especially if they have symptoms. They could be contagious. And so that's how that should be interpreted. But what if the test is negative? The easy thing to do is just default to, oh my God, great, the patient doesn't have COVID-19. But there's really important factors to consider, especially when you're evaluating any manufacturer. There are risk of false negative results depending on how the specimen was obtained, right? So I'm sure I don't know how many of you all have ever had a nasal pharyngeal swab for a flu test. I mean, they have to go pretty far back to get a adequate specimen. How was the specimen handled? Has it been sitting out for a long time? Some of the things that are important to know about the test, especially for the lab, is that once the cartridge is open and taken out of the package, it has to be used within 2 hours. And once it's loaded and ready to go, it has to be ran immediately. We don't know what would happen to the sample if the cartridge was prepared and the sample just sat at the bench, right? And so it's really important to understand that because at this time, we're experiencing such a high volume that requires testing that these processes need to be reinforced in order to ensure the best sample to get results, right? And some test limitations, in addition to what I've already talked about the patient that we're testing, the specimen, what is a negative test. I think that those are really the main things that I think are important to know, and that if you have a negative test, I think asking yourself, if the patient has signs and symptoms, why is my test negative? Don't take it at face value. And the implications right now, think about it as pharmacists, especially on the inpatient side, they're inundated with helping with enrolling patients probably in investigational drug studies that probably require a positive test in order to enroll the patient, right? Or even assisting with obtaining compassionate use medication, which also would require a positive test. So a false negative test could preclude access to medication for a patient. So I think it is important if a healthcare provider suspect that the patient should have tested positive for COVID-19, I mean, we should probably be thinking about repeating the test or really exploring why that test is negative. So I think that gives you a highlight, a little bit about me, a little bit about the instrument, a little bit about the SARS CoV-2 test. I want to acknowledge the following people. These are my colleagues at Genmark from marketing as well as medical and scientific affairs that really helped me prepare the content for today's podcast. And I am so excited to answer your questions.
So I think now if you have questions, go ahead and submit them in the chat function, and we'll go over some of the questions that we've prepared beforehand, and as well as any questions from the audience. So the first question is, how were the Genmark ePlex SARS CoV-2 tests able to be quickly developed, manufactured, and shipped within 1 month?
That's a great question. And this is something that I was super impressed with. And I mean, clearly as a clinical pharmacist, this is all new to me, but Genmark began designing the assays in the last week of January, when only a handful of SARS CoV-2 genomic sequences were publicly available. And they were publicly available to facilitate a design. And we designed this multiple assays with multiple genes with the goal to choose the most optimal design, also while factoring the CDC guidance. And ultimately, we optimized 2 designs using the N gene region, and we began sending kits out for research use only testing to generate some clinical sample data while we completed analytical studies internally. So from the assay design to shipping those first kits, it was about a month. It really highlights the capability of Genmark as a company and the technology to quickly design and release assays. I think, no question, right? Our heroes on the front lines are first responders, our nurses, our physicians that are risking their lives. But we also have heroes within our company that have been working tirelessly in research and design to really attack this pandemic head on. And so it's been really great to experience that as a clinician. I still consider myself a clinician, so.
All right, so question number 2. How is the Genmark EPLEX SARS-CoV V2 test unique compared to the other rapid diagnostic tests granted FDA emergency use authorization for COVID-19? And was there a comparison conducted between this test and others?
Yeah, so that FDA EUA website is a really good source, and so you'll see those 29 manufacturers, and you'll also see the laboratory developed tests. I think one of the main things that's different is the sample to answer solution that we're able to provide. As you saw in the previous slides, it's the workflow is streamlined. It's hands-on time, minute and a half, time to result, hour and a half, and so that design is optimal for labs that have low volume and high volume, but not pandemic volume, right? And so one difference is the sample to answer versus a manual real-time PCR. So the technology and the process, that's different. But also the volume that we're able to support is going to be different compared to other larger batch automated platforms. And we've compared our assay to the CDC assay, and currently our customers that have adopted our SARS-CoV-2 test are comparing it to multiple other solutions currently during their laboratory verification process.
All right, so question number 3. How many Genmark EPLEX systems are currently in use, and how will this impact the need for COVID-19 testing? I think maybe that one first, and then we'll move on to the second one.
Okay. So we have over 500 EPLEX systems placed worldwide. Majority of these are actually running our Respiratory Panel multiplex assay. They're running the RP assay as a part of their COVID-19 testing algorithm, but also some of them are also running our SARS-CoV-2 assay. So this is something to even think about. Laboratories are also forced not only to decide which patients get tested first in their algorithm, but which assay gets tested. So do you run a CoV-2 first, SARS-CoV-2 test first? How do you decide? Or do you run the respiratory panel, and if it's negative, then you run a SARS-CoV-2 assay? That has now shifted, and each hospital institution laboratory is doing things a little differently to meet their needs. I don't think there's a right or a wrong answer. I just think that it's important to be aware of your local situation with COVID-19, especially back in February and March at the beginning of this. We were still in our flu season, right? And there was an interesting preliminary study that came out of Stanford that showed about 22% of patients that had a positive SARS-CoV-2 test also tested positive for other respiratory viruses. So it is important to appreciate there are going to be patients that might be co-infected with 2 different viruses. And just because you had, let's say, a negative or a positive RP1 multiplex result, say with influenza, you may consider running a SARS-CoV-2 assay, right? Because there is a chance that the patient could have both, and maybe the treatment, especially the supportive care, might be different.
So we saw in the, oh, sorry, go ahead. So we saw in the news that there was a recent shipment of the research use only test kits to Hong Kong. Can you describe the impact of the test kits that they've had on diagnosis and treatment in the region?
Sure. So we been shipping kits to Hong Kong in early March as a part of the pre-release early access research use only, or RUO program. Several sites have begun running the tests on patients, and now they're exploring opportunities for more near patient testing, and that's all I could really tell you right now.
Okay, for sure. All right, so question 4. What are your predictions for the Genmark ePlex system following the COVID-19 pandemic?
Wow, that's a great question. I think the one thing that's just a shock to me, I still can't believe it, that I am working for a diagnostic company, and that the demand for a respiratory panel assay and SARS-CoV-2 testing has been extraordinary. Our teams at our company have been focused, working 24/7 to help address this public health emergency, which I just was not mentally prepared for. And we, interestingly, Genmark recently received funding to develop a next version of our ePlex RP panel. So I think we're going to call it the RP2 panel, which will include this SARS-CoV-2 target. So it'll include the virus and bacteria RP1, and then with the SARS-CoV-2 target also included. And I think that's super important, as I previously mentioned, that a single-centered preliminary result showed that 22 patients could be co-infected. And so we received this grant to start developing that test. And I think it'll be important as we prepare for the next flu season to be able to screen for all these viruses at the same time, as well as controlling future transmissions of SARS-CoV-2 virus. I think that is the cornerstone in reopening the US, is to provide a solution for this testing issue and control the outbreak.
All right, so question 5. Can you share how you made the transition from clinical pharmacy to industry?
Oh, okay. So as a pharmacy student, resident, I was basically trained, I call it trained, to think about my 5-year goal and my 10-year goal. And that's something that I recommend to all of you, is to really have a vision of where you're going. And when I was at UT Southwestern, I had moved to Dallas to be closer to home. Home is Oklahoma for me, to work as a stewardship pharmacist. And at that point, I was about 3 years out of residency, 5 years out of pharmacy school. And my 10-year goal was to eventually go into industry, but I wasn't sure what that looked like. Usually industry, for pharmacists at least, means to work for a drug company. However, my training in infectious diseases and stewardship, my values have been to optimize patient care, judicious use of antibiotics. And so I had a little bit of a hard time reconciling representing just one drug if I were to switch to industry. And so this opportunity came up. I was recruited through LinkedIn, by the way. So recommend all of you to update your LinkedIn continuously, like you would update your CV, with this amazing opportunity. And it was hard. It was a question that I actually reached out to multiple people that I've met through networking that have either switched to working for a diagnostic company, to someone that's worked with Genmark but is still a practicing pharmacist, and my mentors, to say, is this the right decision? And ultimately, bottom line, I decided that I was actually trained for this position. So a little bit about my background. I did residency at Johns Hopkins, and when I started in 2013, they had just implemented a rapid diagnostic blood culture solution. And all of the residents were required as a part of their on-call to review all positive blood cultures that came off of the assay and provide recommendations and data collect pretty much, but in real time. So that was my first experience. And then my second experience was at Medical University of South Carolina, where I was a stewardship infectious disease pharmacist. And I rounded in the micro lab every single day and also reviewed positive blood cultures and made recommendations. And so those experiences gave me this unique experience where in my job, I'm able to consult with other institutions that might have different resources. They might have different pharmacy models, they might have different patient populations, and really help them work through how to best implement a rapid diagnostic solution in their patient population, knowing their hospital formulary is different, their antibiogram may be different. Yeah, that was a really long answer.
No, that's awesome. Thank you for sharing. So that's the end of the questions that we have prepared. Are there any questions from the audience that you want to submit now? We can ask Dr. Smith while she's still here. All right, so I think there's no questions that were submitted in the chat. So thank you for joining us today. We're working on our next episode, so stay tuned for more information. We're really excited that Dr. Tiffeny Smith was here to share her expertise for Genmark Diagnostics. And Paula posted a post-discussion form in the chat, so you could fill it out for us. Let us know what you want to hear at our next episode. Thank you.
Thank you so much.
Yeah, thank you so much for joining us. Have a good day.
Bye.